Hello students, today we are going to study ecological adaptations, specifically in halophytes. I am Dr. Akshita Dhaliwal from Department of Botany, Multan University College, Patiala. First of all, what are halophytes? These are special type of xerophilous plants grow on saline soils with high concentration of salts like NaCl, MgCl2 and MgSO4. The soils are physiologically dry and the osmotic pressure values are very high. These are mostly succulents. The leaves are evergreen, thin, small and leathery with water storage tissues. Thick cuticle and well-developed palisade tissues are present. Most of them produce a special type of branched, negatively geotrophic roots that came out of the mud surface to encourage the entry of oxygen gas. These are called as pneumatophores. These pneumatophores are breathing pores which are for gaseous exchange. Some of the halophytes also shows vivipary. Vivipary is the germination of seeds before they shed from parent plant. The universal feature of these plants is selective absorption and tolerance of sodium chloride and the ability to grow in soils so that saline and they are either physiologically toxic to normal plants or impose serious osmotic water stress. So mainly halophytes are divided into four categories according to Warming in 1909. These communities are lithophilous, psamophilous, philophilous and helophilous. As the name indicates, lithophilous are growing on rocks and stones, psamophilous on sand, philophilous on mud and helophilous on swamps. Thus, helophilous are further divided into two, with salt swamp or desert swamp and littoral swamp forest. The most common example of halophytes are Rhizophora and Sonartia. So we are going to study the anatomical characters of Rhizophora mucronata. The roots of this plant is divided into two types, aerial and subterranean. And we are going to understand the vegetative organ of this plant through the tiers of subterranean roots. Its several layered cork cortex is made up of star-shaped or stellate-shaped cells which are connected by each other by lateral arms. Cortical cells have peculiar thickening ridges which are lignified and filled with oil and tannins. The pith cells are thick-walled with pitted thickenings of lignin and contain tannins and oil. This is the diagram showing TS of old subterranean portion of stilt root of Rhizophora mucronata. Next is stem. The young stem of Rhizophora shows very thick cuticle. Epidermal cells are heavily thickened with squarish or cells are filled with oil tannins. Then epidermal cells are, after the epidermal cells, there is hypodermis which is several layered with thick walled cells. Primary cortex contains number of lacuna, thick cells filled with tannin and oil, and some cells possess calcium oxalate crystals, and there are present egg-shaped thickened spicules in primary cortex region which provide mechanical strength to the cortex. The inner cortex contains number of branched thick-walled cells, that is sclerites, which provide mechanical strength. Pericycle is several layered sclerenchymatous It possesses lacuna and S-shaped spicules, and it also contains tannins, and vascular tissues are well developed. This is the PS of young stem of Rhizopora, which shows presence of spicule, lacuna, oil cells, tannin cells, sclerites, endodermis, and all the important points which I earlier told you. Then last is the leaf. In the leaves of Rhizopora, cuticle is very thick and well developed. Epidermal cells are thickened and contains calcium oxalate crystals. Stomata are sunken and present only to lower surface. Several layers of thick walled cells present below the upper epidermis which are filled with water, and the outer layers of this layer filled with oil and tannins. Cells of lowermost cells are mucilaginous that remain embedded into pensil tissue. Mesophyll is well differentiated and porphyry areas are present on the ventral surface. So this is all about the ecological adaptations in halophytes. Thank you.